Hi, everybody. This is Chapter 9 of Concepts of Biology for Biology 160 at Clark College. So let's dive right in. This PowerPoint is about molecular biology, specifically DNA and RNA and protein synthesis, which is the sort of underpinning principles of molecular biology. So how do molecules interact with other molecules in order to get life done? Okay, so starting off, we're going to talk about the purines and pyrimidines. So these are the nitrogenous bases that are part of the nucleic acids we'll be talking about today. Sorry, I'm just going ahead and picking some colors. Ooh, I have more colors now. I upgraded. So here we go. Okay, so remember that nucleic acids are made of nucleotides. That's the name of the molecule. So this whole thing is a nucleotide. I want a smaller pen. There we go. So the nucleotide has some parts, so it's not homogeneous across the board, so there are different pieces to it. So nucleic acids have one or more phosphate groups, a pentose sugar, pentose meaning five-sided, and some kind of nitrogenous base, which is called that because at some of the corners there is a nitrogen and not a carbon. So that's the basics for nucleic acids. Now with ATP, there is an adenosine for a base, and then 3 phosphate groups instead of just 1. And recall that ATP is the energy storage molecule. But now we are switching gears, and we're going to talk about information storage and transmission. So that is the topic today. Okay, so in DNA, which this is, how do I know? Well, if I go, oopsie. If I go look, the sugar here is deoxyribose. That's because there's no O here. So if it was ribose, there would be an O, but this is deoxyribose because there isn't one. So it's unoxygened. Okay, so in DNA, which we've established, there are 2 kinds of nitrogenous bases. So you may be wondering, what do you mean 2 kinds? There's 4 there. Well, what I mean is there are 2 sort of species, and then within those, there are different versions. So the pyrimidines are the single ring structures. So these are cytosine and thymine. So you can see they look pretty similar to each other. They have a ring structure with some hanging offy bits. And then we have the purines, which have 2 rings. 1 ring, 2 rings. So that distinguishes the purines from the pyrimidines. So within DNA bonding, one of the first rules is that purines pair with pyrimidines. So that's the rule, and then we'll establish some more rules a little later on. Okay, so let's look at the pairs interacting with each other. So in DNA, because it's double-stranded and there's a center region, we call different parts of the molecule different things. So it's a double helix, so it looks like a sort of a twisted up ladder structure. And you can see that right here. Yeah, and EpicPen is wanting to tell me to hold shift to draw a square, so I guess we're in tutorial mode. So here we have the double helix of DNA, twisted up ladder, and you'll notice that there's some edge pieces. So the ribbon-like structures on the edge here. Oops, I'm drawing squares again. I meant to switch to pen. All right, so this is the sugar phosphate backbone. So why is it called sugar phosphate? Well, there's a pentose sugar, and then there's a phosphate. So the backbone is an alternating pattern of sugar, phosphate, sugar, phosphate, sugar, phosphate, onward into oblivion, and that's why it's called that. You're also going to notice that phosphates are charged. So they have either, depending on what they're bound to, either a charge of 1 minus or even 2, right? And in the case of DNA, that brings us to the 3' versus 5' end. So without going into too much molecular detail about it, you'll notice that on the sugar phosphate backbone, one end has a hydroxyl group hanging off of it that is attached to the deoxyribose So that's this is the D part of the DNA. And the other end has a phosphate group with 2 negative charges, 1 and 2. This is what determines the 3' versus 5' ends. Now, you don't need to know why they're called that, but those terms are going to be coming into play when we start to talk about replication of DNA and the direction that it proceeds in. So just keep those in mind. But that's how you identify the 3' or 5' end. And this really matters because there's some rules about the way DNA is structured that depend on the 3' and 5' ends as well. All right, so let's look at some other features of this. Here we see some bases interacting, so they're going to interact at the center of the molecule. And one thing that you might notice is that we have, like I promised, our pyrimidines, let's write those PY, are only interacting with purines, PU. Also true is that A always goes with T, adenine and thymine, and C always goes with G. You'll never see an A paired with a G or a C paired with a T. Those are not the way those things interact. You might also notice that the bases interact with each other via hydrogen bonds. So there is a weak attraction between the partially charged H here on this amino group and the O here. And there's a weak attraction between this nitrogen and this hydrogen. So this is a piece where you can prove to yourself also that water isn't the only thing that hydrogen bonds, and it's not always oxygen and hydrogen bonding with each other. So in this case, there's a partial charge on nitrogen, which attracts it to this hydrogen. So individually, hydrogen bonds are very weak, but collectively they're very strong. So although that they seem like they might not be the best choice to bind a DNA molecule together, because of their collective strength, it's actually great. And it creates a situation where when the strand needs to be read, you have to peel the molecule apart. And having hydrogen bonds here instead of covalent bonds makes that easy and relatively inexpensive from a caloric perspective to do. So that is the basics of DNA. I also want to acknowledge that people who discovered this ATCG pairing rule are Watson and Crick. Those were the scientists. And then the person who discovered that DNA was this shape is named Rosalind Franklin. So there are some pioneering scientists we have to thank for this knowledge. They pioneered the work to figure out what DNA was and what the rules for pairing were. Okay, so let's move on here. I already explained this, so I won't spend too much time on it, but I just want to point out, let's get an appropriate color here. Okay, sorry, I had to cough. Didn't want to cough into the mic. So ribose has hydroxyl groups at the 1, 2, and 3' carbons. These are just numbers for how you go around the ring. And deoxyribose has a hydroxyl group at the 1 and 3, but the 2' carbon does not have an oxygen on it. So it just has an H. That's why it's deoxy, because the oxygen has been taken away. It's not there. Alrighty, so when we're talking about DNA and the manipulation of it, we have some things to keep in mind from the perspective of taxonomy and evolutionary history. So just as a reminder, in prokaryotes, there's something called a nucleoid. And it's not a nucleus because there is no membrane. So oid means nucleus-like. As you can see, it's just kind of a wad of DNA in the center of the cell. Eukaryotes, by contrast, have a double membrane system. So there's an inner and an outer membrane that encloses the space that contains the DNA and protects the DNA. So there are some differences in the way DNA is dealt with between prokaryotes and eukaryotes for that reason. There's an extra barrier in the way for eukaryotes where there is not for prokaryotes. Another difference is that eukaryotes just straight up have more DNA. So they have a very, 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 very long collection of molecules, so the DNA is not one continuous molecule, it's many actually. And so in order to effectively keep that wrapped up in a way that's manageable, and also in preparation for cell division, which we'll cover in the next video, some organizational stuff has to happen. So if you have a long molecule, think of it like yarn or rope. If I hand you a wad of tangled up rope and say, I'm going to need you to divide this exactly in half for me, and make sure it's exactly in half, not 49.5%, etc., but exactly in half. You'd have a hard time doing it if you didn't first organize the rope, right? So the same principle applies to eukaryotic DNA. So in each of these rectangles, this one is smallest, so we're zooming out. So this is us zooming out from little to big. So the DNA gets wrapped around a histone, which is this, and a histone is a 4 subunit, that's not how you spell unit, a 4 subunit protein. And they basically function as spools, as for thread, for example. So the DNA wraps around the histone, and then there's a series of histones, and each histone has the DNA going around it about 1.5 times. And then the next histone comes along. So what you get is sort of like this beads on a string looking thing. So the name for a histone plus the DNA that's around it is a nucleosome. And those end up coiled into what's called a chromatin fiber, which is this entire thing. Those condense further to form chromatin, which is the substance that is found in the nucleus of eukaryotic cells. And then when it comes time to duplicate the DNA in preparation for division, all the DNA gets copied, and the copied DNA is organized into these X-shaped structures called chromosomes. So that's how those work. Okay, so the next concept we need to understand is complementary and antiparallel. So complementary and antiparallel. Oh no. Sorry, that was the PowerPoint version where the weird spike things come out occasionally when I'm drawing, so I'm going to use Epic Pen instead. Oh, and by the way, if anybody ever wants to make videos like this for any reason, be it teachery or not, the software I'm using is called Epic Pen. There's a free version. I have the pro version, which costs about $20. But you can do whiteboard and blackboard stuff with it, and there's a bunch of other really cool features that allow you to write on the screen. And not only like PowerPoint, but I could just, like, if I was making an instructional video for how to do a computer thing, I could literally just write on top of my screen using this software. It's really awesome. So if you want that, there it is. Okay, so 2 strands of DNA are complementary, meaning that the sequence of bases in one strand can be used to create the correct sequence of bases in the other, and that's due to the pairing rules. So because A always goes with T and G always goes with C, if you see an A, even if you can't see the other strand, you know that a T must go there. There's no other choice. So that imposes order. So the complementary base pairing rule is what dictates this, and it's one of the most powerful and important concepts in genetics. So it's a really big deal. And the way that they are annotated is by putting the number of colons that corresponds to the number of hydrogen bonds that bind them. Okay, so the 2 strands of the DNA molecule are also running anti-parallel to each other. So they're running in opposite directions. So you'll never see 2 5' on the same end. Instead, they run the opposite. So the 5 and the 3 are reversed on the 2 strands. And that helps to determine which of the 2 strands is going to get copied continuously and which of the 2 strands is going to be the lagging strand, which is a principle we'll discuss momentarily. So in a moment, we're going to look at the specifics of DNA replication. So in order to successfully divide, any cell that's planning on dividing has to copy all of its DNA first, and that's the reason for that 50-50 split. But when you introduce copying DNA, depending on which strand you read, etc., so forth, there's a couple different possibilities. So one would be that you could open this up here and read whatever sequence is on this strand. So let's say I'll make a model strand. It is T, A, A, T, G, C. I'm just going to keep it short and sweet. So if I opened up this and I read this strand, I could add the appropriate nucleotides, right? And then I could abandon this strand altogether and make another corresponding new strand. And that would mean that this DNA molecule that I've just made is brand new in its entirety, so it contains no components of the old one. But that's not how DNA replication goes. Instead, there are two strands. Both strands are copied, 1, 2, such that each daughter DNA molecule, which is what these two guys are, is half new and half old. So it's called semi-conservative because half of the original DNA molecule is conserved within each daughter molecule. And if you want to watch the DNA replication video, you can click that, but we're not going to do that today because I'm already making a video. A video within a video would be very meta, however. Okay, so I mentioned that there's a leading and a lagging strand, meaning that the strands are not synthesized in exactly the same way. The result is the same. The daughter molecules that come out at the end are going to be identical to each other, but because of the 3' and 5' directionality and the antiparallel nature of the strands, they have to be made a little differently, and that's because the enzyme that does all of this is called DNA polymerase, and DNA polymerase can only go in one direction. So specifically, it likes to synthesize DNA from its 5' end towards its 3' end. So it likes to bind to the 3' end of the leading strand. So the leading strand is also synthesized continuously, right? So you can see that this template strand and this have no breaks in them, unlike what's going on over here. So let's zoom out and go back to basics really quick, and we can look at what's happening here. So I mentioned that in order for DNA to get replicated, it needs to get unwound, and that's one of the reasons that having hydrogen bonds in the center of it is useful. So that begs the question, if you want to unzip the DNA molecule, you first have to unhelix it, so untwist it, and then unzip the untwisted part, right? So that requires some enzymatic action. So there's some enzymes called helicases that do the unwinding. Then we've got some single-strand binding proteins, and they sound like, so they only bind to single-stranded DNA. And what they do is they sort of stabilize the opened bit of DNA so that the fork stays open and has structural integrity. So the replication fork is just the name for the literal fork in the DNA road that is created when DNA is unzipped. Now, as we said, DNA polymerase is going to be actually doing the work of polymerizing the DNA molecule, and it likes to synthesize things in the 5' to 3' direction. So that's going to do that for the leading strand, but the lagging strand has the opposite thing going on, right? So if it likes to synthesize things from 5' to 3', and we know that this is the 5' end, so this must be the 3' end, it has to go this way. It can't go this way. And that creates some special problems because of the way that DNA polymerase works. So it can't just continuously go from the replication fork out to here. It has to do something a little different. So there's another enzyme that works here called primase. So it's an ACE, which means we know it's an enzyme. And prime suggests that it's going to prime something. And indeed, what it does is it places an RNA primer. And the RNA primer provides the following opportunity. So the RNA is going to be also antiparallel and complementary. So it's going to provide a 5' end for DNA polymerase to work from, because remember, it likes to synthesize in the 5' to 3' direction. So the 5' end of the RNA primer is where DNA polymerase can land and start from there. So these little fragments of DNA we call Okazaki fragments because that's who discovered them, and they eventually meet up with each other, like what's happening here, and they are joined together by an enzyme called DNA ligase. So ligation, or to ligate something, means to join two things together, and DNA ligase just glues the Okazaki fragments together until we have a daughter molecule that is identical to the other one that's being produced. All right, so here's another picture of the same thing. I'm not going to do a bunch of drawing on here because we already did that, but you can see that the discontinuous nature of the lagging strand is preserved here. And also that if you have an opened up bubble of DNA, which is the leading and which is the lagging are going to vary depending on which fork you're at, right? Because they have antiparallel designations here. So 3 and 5 versus 5 and 3. So this is what the replication forks as a whole look like. Okay, so enzymes are great, but they're not perfect. So sometimes enzymes can make mistakes, or sometimes mistakes just happen. So one of the mistakes that tends to be made during the replication of DNA is that either a wrong base is added, or some things bind together that they're not supposed to. So let's start with the first example, and that is mismatch repair. So here we see a G and a T, and this creates a bubbling out of the sugar phosphate backbone here. That's because those two aren't supposed to interact, and they don't, so they make a little bulge in the molecule. This bulge can be recognized by DNA polymerase and fixed. So that's what we mean by proofreading. It looks at the, and I'm using the word looks in air quotes here, not that you can see me, it looks at the DNA molecule and says, oh golly, those aren't supposed to be there, and it will snip out the offending non-match and replace it with a match. The other kind of repair that happens is called nucleotide excision repairs. So this often happens when you have pyrimidine dimers. So what you have here is two thymines, and remember, thymine and cytosine are pyrimidines. They're the ones with one ring. So pyrimidine dimers tend to form, and these are covalent bonds between two neighboring pyrimidines, and this is bad. This is not how this is supposed to go. These guys are supposed to be bound to the A's across from them, not to each other. So in nucleotide excision repair, what happens is some restriction enzymes, which are able to identify the place to chop, will chop out the offending chunk, and usually some bases on either side, and then replace all of it with something that actually does match. That's called nucleotide excision repair. And it's different because it takes out neighboring pieces of the strand instead of simply replacing the base. Okay, so the ends of linear chromosomes are naked, and that puts them at risk for damage, right? So if you have all of this important information in here, the stuff that's on the end is going to be more prone to be damaged. So what do we do about that? Well, there's a bunch of non-coding DNA at the ends of our chromosomes called telomeres. So these are areas that don't code for anything, and they're end caps that are protective in nature. So they protect the DNA, the chromosome ends rather, from damage. And as we age, I'm using we royally here, so as eukaryotes age, with very few exceptions, our telomeres get shorter. So the shorter and shorter your telomeres get, the more signs of aging you will show. So the telomeres need to be maintained and improved in order for cell health and DNA health to be good. So there's an enzyme called telomerase that does this, and this is Elizabeth Blackburn. She discovered how this worked and won a Nobel Prize for it. Way to go. So what telomerase does is it has an associated RNA molecule that provides the primer from which to build additional telomeres. So it's kind of like primase in DNA replication of the lagging strand, but instead it uses RNA to prime to add more nucleotides in the telomere. So it's not replicative in function, but rather replacement. So basically, telomerase has this DNA or RNA sequence, which provides a starting point for telomerase to add more nucleotides. And it does this first to the leading strand. So you can see this is being extended that way. And then after that, primase and DNA polymerase are going to do the rest of the work. So telomerase only elongates one strand and then lets DNA polymerase do the rest of it. But together, what they accomplish is a DNA molecule that's a little bit longer than it was before. So you guys see that? So up here, so we've added this much more telomere, which is good. It's protective. So the telomeres are those caps on the ends of chromosomes. They shorten as we age. And remember, I said in lecture, there's a lot of scientific work around senescence and aging. And senescence, by the way, is the name for the part of aging where not only do you get older, but your body gets crappier. So a lot of that work concerns telomeres and how to maintain them. So Dr. Blackburn was doing some of that work when she figured out about telomerase, which is a really big deal. And some more recent telomere news is that Scott Kelly, the astronaut who spent an entire year in space, when he came back after testing a bunch of things about him, NASA scientists discovered that, hey, his telomeres got a little longer. Why? We're not really sure. We just know that they did, which is super interesting. Okay, so let's talk about the central dogma of molecular biology. So the central dogma is this principle that states that DNA encodes RNA. So you can infer what the RNA sequence will be by looking at a DNA molecule. And RNA in turn encodes protein. So these are the steps that one must take to perform protein synthesis. And we're going to talk a lot about this and exactly how it's done. And in fact, we've already done a bunch of this in class. So the things I expect you to take away from this presentation, in addition to rules about the shape and structure of DNA and why everything is the way it is you need to be able to walk me through the production of a protein from the events in the nucleus to the events in the cytoplasm or on the RER, and what happens after that as well. So knowing how the stuff works is important, but also being able to identify the location in a eukaryotic cell where it's going on is important, as well as what enzymes are doing which tasks. So when I talk in lecture about you needing to have deep knowledge in order to successfully answer exam questions, this is one of the things I'm talking about. Okay, so there is transcription and translation, and these are different. So transcription is the act of taking some DNA and making an mRNA transcript out of it. So why is it called transcription? Well, if I transcribe someone's notes, I'm just going to write the notes word for word in the same language. So we're going from nucleic acid language to another nucleic acid language. Translation, by contrast, takes a nucleic acid language and translates that that nucleic acid language into amino acid language. So we have a language switch. So that's the reason for those names. They also have different locations. So transcription always happens in the nucleus. And translation, because it requires ribosomes, happens anywhere where there are ribosomes. So we know from our studies of cell biology that those places include the cytoplasm or the rough endoplasmic reticulum, one or the other. Okay, so let's talk about transcription. Now, if we know that DNA polymerase is an enzyme that polymerizes DNA molecules, it makes sense that RNA polymerase is going to do the same task, but it's going to create a complementary strand of RNA rather than DNA. So helicase unwinds the DNA, just like in DNA replication, but instead of having DNA polymerase land there, RNA polymerase does instead. And there's a bunch of other proteins involved as well, but we're not going to go into those specifically. So we call those the transcriptome. Which is the name for the sum total of all of the enzymes and other proteins that are involved in transcription. And they bind at a region called the promoter, which is not going to be part of the RNA transcript, but rather the place on the DNA where everything touches down from the transcriptome. And it's the sequence that says, start here. So RNA polymerase makes a copy of the gene to be produced, and this is called the messenger RNA, or mRNA. So mRNA is the one that is going to be translated into a protein. So here's another view, and this is our first glimpse of one of the differences between DNA and RNA in addition to the ribose-deoxyribose difference. Note that on the RNA molecule here, you do not see any T's. We have U instead, and U is uracil. So DNA has thymine, RNA has uracil. So if you see a U, you know it's RNA. Other than that, they're the same. So they both still have a 5' and 3' end. That's why RNA can be used as a primer for DNA polymerase. And they have C's and G's and A's. It's just that the T is gone, U is there instead. Okay, so once the RNA transcript is made, we're not done yet. We can't quite send this thing to a ribosome yet. So one cool thing about the human genome, and eukaryotic genomes in general, is that one gene, so one region of DNA, can code for multiple proteins depending on how you chop it up. So you have lots of opportunities within a single gene to do different things with that gene depending on how you chop it. So the primary RNA transcript is the name for the RNA that's fresh off the DNA. So you can think of it as being kind of like hot off the presses, so to speak. But RNA undergoes processing on its way out of the nucleus so that the finished RNA looks a little bit different. So two things happen. One of those things is that some end pieces are created. So we have the 5' cap, and on the 3' end, we have the poly A tail, which just looks like somebody saying, ah, which I think is funny. But I'm a nerd. What can I say? We also have some flanking untranslated regions, and these vary in length and character depending on the RNA. But notice the introns are the pieces that are spliced out. So where we before had exon, intron, exon, intron, exon, now we have only exons. So the exons are the ones that are going to be preserved, and the codons in there are going to code for amino acids. So mRNA uses introns and exons, and introns are like intermission. There's no information in them, so they get cut out. Exons enable some portions of the code to be used while others are discarded. And then the caps are put on both to protect the mRNA transcript because it contains valuable information, but also to provide a cue for where on the transcript the ribosome is to begin. So introns are cut out and discarded. They're the intervening sequences. And exons are retained, spliced together, and are expressed. Okay, so we have alternative splicing. This is just a demonstration to show you how many different possible gene products you can make from the same, the same mRNA. So these are all primary mRNAs over here. And these are the possible gene products. So let's count here. We've got 1, 2, 3, 4, 5 primary mRNAs, and for each of those, one or even sometimes greater than one possible gene product. So we've got 10 here. That's a lot. So you can store and change a lot of information just using this technique. So I'm not going to expect you to memorize all of these definitions and what they look like, But rather, this slide is just kind of to prove the point that, hey, alternative splicing can produce a staggering variety of proteins, even from the same or similar mRNAs. So that gives our genome a lot of latitude. And in fact, if we couldn't do this, and we had to have a completely separate gene for every single protein, our genome would be a lot longer. So our DNA molecules would be longer, and we might have more chromosomes. So that's something to think about. Okay, so we're going to go back to eukaryotes just briefly. And that's because due to the lack of a nucleus, as I foreshadowed earlier, they have some differences in the way that they do the whole protein production thing. So mRNA comes from DNA, and you can see that here. So it's sort of extruding off the side. And you can infer from that the, So RNA polymerase must be here. And then on each transcript, as it's extruded from the DNA molecule, ribosomes bind to it, and these are going to make amino acid sequences. So what this means is that inside of a bacterium, protein production can happen really, really quickly, and proteins can rapidly reach a really high concentration inside the bacterial cell. And in fact, because of course we know that protein is the workhorse of a cell, it's the macromolecule kind that does the biggest job, if you want to kill or disrupt a bacterium, a good way to do that is to prevent it from making any protein. So some antibiotics act to inhibit RNA polymerase, which prevents bacteria from making the proteins that they use to harm us. All right, so we're going to go ahead and skip this. It's a good one, but you can watch it on your own time. And we're going to finish up by talking about translation at a ribosome. So remember, translation is taking the language encoded in the mRNA and turning it into a language of amino acids instead. So we're going from nucleic acids to amino acids, and that's why it's called translation. So the ribosome has a large and small subunit, and these clamp around the mRNA molecule such that tRNAs can bind and bring the appropriate amino acid to the growing chain. So translation begins when a tRNA anticodon, and let's circle that, recognizes a codon on the mRNA. So this is the codon. Okay, so these pieces of terminology are important because if you don't understand them and I ask you a question on the next exam about them, you won't be able to confidently answer. So make sure you've got your definitional terms down. I was kind of noticing a little bit of that when I was grading the last exam. It seems like some folks were missing some key definitions which were required to understand what the question was asking. So if you don't know the terminology, you can't understand what the question is asking, and it's really hard to be successful on the exam. So make sure that you're getting your vocabulary in there as you're studying. So anticodon, tRNA always. Codon, mRNA. Okay, so we have the small ribosomal subunit down here, and then the first tRNA is going to land on the start codon. So AUG stands for methionine. It codes for that. And it also is the begin translating here codon. So we call it the start codon. So we can see here's methionine being carried by this tRNA. And the tRNA has an anticodon that is complementary to AUG, so UAC. There was some confusion in lecture about where the tRNAs come from. You don't actually need to worry about that. Of course, they do have to come from somewhere. They are made and then they basically cycle through doing this and then getting recharged and picking up another amino acid. So they're going to bring the amino acid to the ribosome, but they're not made from the mRNA and they're not made, well, they're made from DNA, but not in the same way. So don't get mixed up and think that somehow the tRNA comes from the mRNA. That's not the case. Okay, so once the methionine carrying tRNA has landed, that causes the recruitment of the large ribosomal subunit. And it permits a second tRNA to arrive. And the reason we're being vague about a second tRNA is because which one it is in particular depends on what the next codon says. So the complementarity between the mRNA and the tRNA is what controls the order of amino acids that are added to the chain. So in this case, the next codon is UUU. And I happen to know, just from doing this long enough time, that codon codes for phenylalanine, and we'll see a picture in the slide I accidentally just revealed. So the corresponding tRNA is going to have the anticodon AAA, and that carries phenylalanine to the chain. So notice now we have letters in here as well, so there's P and A. These aren't designations for the tRNA, but rather for the binding site within the ribosome that they stick to. I don't really like the way they've drawn this here because it implies that the P and the A are some kind of feature of the tRNA. That's not true. So the P site is the first binding site, so every tRNA is first in the A and then in the P site with the exception of the first one. So you can see phenylalanine is going to come here and land in the A site, but then once the tRNA carrying methionine has left, it's exiting, having contributed its amino acid, it's going to go away, and now the phenylalanine carrying tRNA is going to move into the P site, permitting a new tRNA to occupy the A site. So let's move on here. So the entry of a release factor into the A site terminates translation and the components dissociate. So there are a variety of release factors and they are coded for by the stop codon. And once the ribosome encounters the stop codon, the release factors come into play and the whole ribosome basically dissociates. So that's what's happening there. Okay, so I accidentally revealed this earlier, but here it is. So this is the mRNA codon chart. So what it tells you is which sequences on the RNA code for which particular amino acids. So here's how you use this chart. You'll notice that the edges say first letter, second letter, and third letter. So when you're reading an mRNA sequence, what you're going to do is say, well, oh, gosh, I wonder what that codes for. What amino acid does that mean? So let's say, for the sake of easiness, purely, that it is phenylalanine. So phenylalanine is UUU. So let's say I didn't know that already, and I saw UUU all in a row and said, I wonder which amino acid that is. I would use this chart as follows. So I would take the first letter and be like, okay, so U. So here is the row that I should be in. And the second letter is also U, so that tells me which column I should be in. And then the third letter has this little key here, so I know that I want to be in the top of the stack, which gives me this. What if instead it was UCU? Well, then I would do the following. Same steps, different order. So I would say, okay, U, I want to be in this top row. C, I'm going to be in the second column. And U, again, top codon. That codes for serine, not phenylalanine. Also note, and this is the last thing I'm going to say in this particular video because we're just about at the 45-minute mark, which is the upload limiting factor. So notice also that the code is redundant. So UUU codes for phenylalanine, but so does UUC. And for serine, you got a bunch of choices. As long as it starts with UC, doesn't matter what the end one is, still serine. So what this means is that this code is redundant. So there's more than one possible codon for each amino acid. And that's helpful because what it means is if there's a single change in the particular amino acid sequence, or excuse me, the particular mRNA sequence, like let's say due to a random mutation, this C became a G. But UCG still codes for serine, so no big deal. Even though there's been a change in the genome, there hasn't been a change in the amino acid sequence that's produced from that genome, and that protects our genome from having random mutations really screw up the works. Sorry, I don't know if you can hear in the background, but my dog is having a dog episode. So that's how you use the codon chart, and it's also one of the significant features of our genome is that it has built-in features to protect itself. So in the next video, I'm going to finish this PowerPoint up, and I'll probably upload some Khan Academy style tutorial stuff that just kind of recapitulates what we did in class with the activity where I asked you to go from DNA all the way to amino acids and vice versa, so you can sort of practice those skills. So thanks for your attention, and I'll see you in the next video.